blood clotting. When we have a broken blood vessel, we need our blood to clot so we don't bleed to death. Blood clotting is also known as hemostasis, not to be confused with homeostasis, right? Hemostasis, heme means blood, stasis means to stay. It's literally translating into keeping your blood in your blood vessel. Blood stays, right? It's a very effective means of closing breaks in small blood vessels, right? You can't form a clot when there's a really big break, as we well know. In order to have clotting to occur, we need certain ingredients, right? First of all, we need a special cell type called a platelet. Some people will also call these thrombocytes. So thrombocytes and platelets are the same thing. Platelets are derived from the same hematopoietic stem cell as white blood cells and red blood cells. When certain chemical ingredients are present, the hematopoietic stem cell differentiates eventually into what we call a megakaryocyte, which is the cell that is going to produce platelets. Platelets are weird, kind of like how red blood cells are weird, right? Red blood cells didn't have any organelles inside of them, right? They were just bags of hemoglobin. Platelets are similar to red blood cells, and they're just little bags of stuff, but they're very tiny, and they're not full of hemoglobin. They're full of things that are important for the process of clotting. And the way a platelet is formed is kind of interesting. The megakaryocyte, and this cell is huge. It's like a giant in the bone marrow. It sits in the bone marrow, and little pieces of its cell membrane and cytoplasm just kind of blip off like little vesicles into the bloodstream, and then we call those platelets. So some people like to argue whether or not platelets are really cells, but we're going to call them cells. Other than platelets, we need certain chemicals in order for clotting or hemostasis to occur. There's a whole group of chemicals that we're just going to collectively call clotting factors. In addition to that, we need calcium and some vitamins, and very specific certain proteins are needed for this process, like prothrombin, thrombin, fibrinogen, and fibrin. So now let's take a look at the steps of hemostasis. There are three major steps in hemostasis: vascular spasm, platelet plug formation, and then coagulation. Let's take a look at each of these in a little bit more detail. Vascular spasm. Vascular means vessel, as in blood vessel. So with vascular spasm, what's happening is the muscle in the wall of the blood vessel is spasming or contracting. And this is important because we're going to get contraction of the blood vessel at the area of the break, and that's going to make the lumen of the blood vessel get smaller so that less blood is coming to that area of the break and less blood can actually leak out of that blood vessel. So this is an important beginning step for blood clotting. Step 2 is platelet plug formation. So you can kind of think of this as the beginning mini version of a clot, okay? When we have a broken blood vessel wall, there's going to be sticky outy pieces. Like when you look at a skyscraper, right? A lot of times, especially those glass ones, they look nice and smooth, right? And that's how the inside of our blood vessels are most of the time. They're nice and smooth. There's no bumps there, right? But if that building were to break, right, there'd be all kinds of like metal pieces sticking out and glass sticking out, and there would be sticky outy pieces. The same thing happens when we break our blood vessel wall. There's all sorts of tissues and fibers that are sticking out, and platelets love, love, love to stick to those sticky outy pieces, right? They won't stick to smooth surfaces, but anything bumpy, they're going to grab onto. And as soon as the platelet grabs onto that rough surface, it activates. And once it's activated, it's releasing some of the chemicals inside of it. Those chemicals are going to attract more platelets to the area. So more platelets will come in and clump up with those other platelets and release more chemicals that will recruit more platelets, so on and so forth. Think back to Bio 271. Was that an example of negative feedback or positive feedback? Our set point for clotting under normal circumstances is none, and we're going away from set point here. So this is an example of positive feedback. third step in hemostasis is the most complicated, and this is the actual clot formation step called coagulation. The platelet plug is a nice handy start for blocking off blood flow to that area where there's the break, but we need something that's a little bit more, I don't want to say permanent, but it's a little bit more permanent than the actual platelet plug. This clot will eventually be dissolved, but it's a lot stronger than the platelet plug, and it's going to block blood from flowing out of the blood vessel until repair processes can occur to repair that blood vessel wall. So coagulation is the formation of the blood clot, and like I said, it's way more durable. That's a better word, not permanent, but way more durable than the platelet plug. See, I should look at my slides. So the coagulation step is pretty complicated, but I'm going to narrow it down to certain specific topics. The whole goal of the coagulation step is to form a sticky net-like trap that will form the clot. The basis of the sticky net is a protein called fibrin. And fibrin is the active form of a blood plasma protein called fibrinogen. So in order to get this net, we have to activate fibrinogen. The way we do this is to use another protein called thrombin. But thrombin itself is inactive in the blood plasma in the form called prothrombin. The question becomes, how do we activate prothrombin? Well, that's where these chemicals coming out of the platelets come in handy. So the chemicals released by the platelets will eventually cause prothrombin to be converted into its active form, thrombin. Thrombin will then find fibrinogen and activate that into fibrin. And the fibrin, like I said, forms that sticky net, and basically it traps our blood cells in it. Red blood cells, white blood cells, platelets all get stuck in the sticky net, and that forms the wall that becomes the clot. Now, I've massively oversimplified this process. There's a whole cascade of events. There's multiple pathways of cascades of events that cause blood clotting to occur or coagulation. So we talked a little bit about the fibrin, but all those clotting factors are going to be involved here. Vitamin K plays a really important role with blood clotting. Babies, when they're first born, they're given a vitamin K shot. And that's because most of our vitamin K is actually coming from bacteria in our intestines, and babies don't have bacteria in their intestines yet because they've been in the sterile environment of the uterus. So they get a vitamin K injection so that if something happens and they need to clot, they have that vitamin K present. You've probably heard of anticoagulants. Anticoagulants are chemicals that will stop the process of coagulation, so they stop step 3 in hemostasis. You've probably heard of blood thinners. They're also known as thrombolytics. These are drugs that are used to break up and prevent clots. So we can use this to prevent and treat heart attack and stroke. Now, some of you work in the medical field already, and you probably already just in your head thought of 10 different blood thinners, right? But some of you are new to this. But those of you that are new to this, you probably know a blood thinner, right? You maybe have heard if someone is having a heart attack or stroke, and they can still talk, that you can possibly give them an aspirin, right? And aspirin is a blood thinner, right? So people that have had previous heart and stroke history sometimes are prescribed a baby aspirin by their doctor in order to keep their blood a little bit thinner and prevent unwanted clotting. Because basically that's what causes most heart attacks and strokes is unwanted clotting. Warfarin or Coumadin is another common drug that's used as an anticoagulant. And so this can be used as a drug, but it's really potent. So they do use it, but they don't like to use it for long periods of time. What I kind of find interesting is the use of warfarin or Coumadin for things other than medicine. So here's my question. Why are biologists finding dead hawks and dead owls and other dead predatory birds? And these birds have toxic levels of warfarin in their bodies, right? So basically what this is saying is that these predatory birds died from excessive blood loss. So where are these birds getting warfarin from? Rat poison. It's the main ingredient in most rat poisons. So the rat poison, the warfarin in that causes the rat to die, but then the predatory birds eat the rats, and then they build up toxic levels of warfarin inside of themselves. A really interesting aspect to anticoagulants is medicinal leeches. So I have the question on here, crackpot medicine or effective treatment? Well, like most things, it really depends upon how you use it. So it turns out that leeches, when they latch onto the skin, they release a compound called hirudin. And hirudin is an anticoagulant. And of course, the leech wants your blood to flow right into its body. It doesn't want any clotting to occur. And the hirudin is going to prevent your blood from clotting and get that nice steady flow right into the yummy, yummy leech, right? How can this help in medicine? Mostly it's used effectively in medicine when someone has like a fingertip that has been cut off or maybe part of their ear or something. And when that structure is reattached, they'll put the leeches on the distal end. And what the leeches do is they make sure you get continuous blood flow. So let's say the tip of my finger got chopped off, right? And you put it back on, you put the leeches over here, and they are constantly sucking the blood, causing it to flow into the area, the tip. And this is helpful because a lot of times these reattachments fail because the blood fails to come to the area. So that's one way they can be used effectively. I'm sure there are others, but that is definitely one. Coagulation is the last step in hemostasis, but that doesn't mean we're done yet. There's some other things that have to happen. Clot retraction being one of them. Clot retraction sounds a lot like clot contraction, and that's what's happening here. It turns out that inside the platelets are contractible protein fibers, and when these protein fibers contract, the clot kind of constricts together and it pulls the broken edges of the blood vessel closer together. And if you pull the broken edges of the blood vessel closer together, it's going to be a lot easier to repair that blood vessel. So clot retraction is an important step in getting that blood vessel wall to heal up so that we can eventually get rid of this clot. And that is our last topic on this slide, how we get rid of the clot once the vessel is repaired. And that is where fibrinolysis comes into play. So there's a different cascade of reactions that will occur once the blood vessel wall is no longer damaged. And this cascade of reactions involves a blood plasma protein called plasminogen. Plasminogen is normally inactive, but when we're ready to dissolve our clot, it is activated into its form called plasmin. And I think of plasmin as like a little pair of molecular scissors that are in your bloodstream, and they're going to cut the fibrin strands of that sticky net, essentially getting rid of our fibrin net, and that gets rid of our clot. When we were looking at anticoagulants, I mentioned that unwanted clotting is a common cause of heart attack and stroke, so I just want to talk about that in a little bit more detail for a second. So normally blood only clots in response to blood vessel damage, but unwanted clotting can occur. This often occurs when we have plaques on the inside of our blood vessels, which are like buildups of fat and cells, and those are not smooth and they can attract platelets and you can get a clot. Now there's two types of clotting that can become problematic, a thrombus and an embolus. A thrombus is a blood clot, but it's typically, we use that term when the clot forms in an unwanted location. So if you cut your blood vessel and you get a blood clot because you have a cut, that's just a clot. But if you don't have a cut and you form a clot, that's a thrombus. A thrombus can turn into an embolus if it breaks away and starts traveling through the bloodstream. And this is really problematic because as blood flows through arteries, the arteries get smaller and smaller and smaller. So eventually that clot might plug up an artery, cutting off the blood supply to that organ. And as I mentioned, the main cause of this are plaques. The fancy name for the condition of having plaques in your arteries is atherosclerosis. So sclerosis is scarring. So you have these scarry kind of plaques inside your blood vessels that the platelets like to stick to. So heart attack and stroke can be caused by unwanted clotting, but they have other causes too. My dad had a heart attack and it wasn't from unwanted clotting. we have a genetic condition in our family that we have abnormal arterial blood flow. Our coronary arteries are different, and that was what caused his heart attack and all of his brothers too. So there are other causes, but these are the main. Unwanted clotting is the main cause of heart attack and stroke.